This is Biolagy. We will start our biology lesson, grade 12. Last session we have seen about the definition of genetics. We have said that genetics is the science of heredity. We have also seen that heredity deals about the traits that is transferred from one generation to the next generation. Today we are going to see one of the genetic crosses known as monohybrid cross. Students, can you guess the meaning of monohybrid cross? Very good. I think you have tried. Monohybrid cross is a genetic cross or genetic breeding situation that relates to just one trait or one characteristic. The word mono means single or one. So monohybrid cross is a cross, genetic cross, that's about one characteristic or one trait or one feature. Today we are going to see and take examples of Mendelian monohybrid cross. Can you remember the work of Mendel? As you can see from the screen, yes, Mendel was the father of genetics, the father of this discipline. He was Greek, Australian monk. He have done his experiment using pea plant. He have taken 7 contrasting characteristics or traits. The first one was height or stem height. This could be either tall or dwarf. So you can see these 7 characteristics: flower color, flower position, seed color, seed shape, pod color, and pod shape. These 7 characteristics, he have taken these 7 characteristics. We will take one example of these 7 characteristics and we will focus on flower color. Now let's see how Mendel did his experiment. First and foremost, he removed the stamen from the flower of the purple flower. Then he used a paintbrush to transfer pollen grain from the flower of white flower to the carpel of the purple flower. And then he pollinated carpel, produced pea pod containing several seeds. Lastly, he collected and grew all seeds from all the pods. And when the plants were mature, he noted that the color of their flower. This is the procedure. This is the Mendel's technique. From this picture, you can see that he first removed the stamen from the purple flower. Then he transferred this pollen grain from stamen of white flower to purple flower. In step 3, he pollinated this matured into pod and then planted these seeds. And he lastly examined the F1 generation, first filial generation of springs. In fact, as you can see, all the flowers have become purple in color. You can see that the phenotype or physical appearance of all the flowers become purple in color. Mendel also carried out reciprocal crosses. He crossed the F1 generation, and in F2 he found that the phenotype becomes 3-to-1 ratio. Now from this picture, these are the results of the 7 contrasting characteristics. If we take the first example, flower color, as an example, purple and white, in fact, the dominant trait, the trait that expressed was purple, and the recessive trait was white. He have used in F2 generation 705 purple flower and 224 white flowers. If you divide 705 to 224, you will get 3.15. That means 3.15, 3-to-1 ratio. Every 3 purple color, you find 1 white flower. It goes on axial and terminal flower position. If you divide 651 to 207, you get 3.14 to 1. You can check and divide and check for all his 7 contrasting characteristics. How he explained his result. Every trait like flower color, seed shape, seed color, pod shape, pod color, all is controlled by heritable factor today known as genes. Genes are unit of heredity. So every trait like flower color, seed shape, seed color are controlled by these genes. And genes are made up of in a different alleles. Secondly, if the two alleles in the individual are different, obviously one is dominant and the other is recessive. One expresses itself and the other cannot express itself. The one that expresses itself is dominant. The one that does not express itself is the recessive. In fact, the dominant trait masks the appearance of the recessive trait. Like the purple dominates the white color. The only physical link between the generation is the gamete or sex cells. This must pass the genes from one generation to the next generation. What passes from one generation to the next generation is the genes are transferred by gametes or sex cells. The genes or alleles which genes are composed of separate when the gametes or sex cells are formed. So each gamete contains only one allele. These alleles are the one that controls the characteristic or the trait. This is known as Mendel's law of segregation. It's known as Mendel's law of segregation. He also stated that the gametes or sex cells fuse randomly when there is fertilization. At fertilization stage, gametes or sex cells fuse together. Let's go on and see the genetic base of Mendel's result. Let's take again the examples of flower color, purple flower and white flower. P generation or parental generation are the purple flower and white flower. The purple flower and the white flower. When these flowers cross together in F1 generation, we found that all flowers are purple in color. Mind you that purple have dominant characteristic. It dominates the white flower or white color. When F1 generation or F1 generation result cross together, then the result of F2 generation will become 3/4 of the plants will be purple flower, while 1/4 of the flowers will have only white in color. This is phenotypic or phenotype 3:1 ratio. 3 flowers purple in color, only 1 flower white in color. This is physical appearance. But if we see the genotype, one of the flower is fully purple, capital P, uppercase of P, P, and 2 flowers will be P, small p, and 1 flower would be small p, that's white color. So the ratio would be, when you see the genotype, 1 to 2 to 1. You can see here in a detail the genetic makeup of these 2 flowers. If we take the purple flower as capital P, uppercase, and white flower, small p, lowercase, the gametes would be P, capital P, and small p. F1 generation would be all PP. That is 1 uppercase and 1 lowercase, 1 capital, 1 small. Still the gametes, when we cross F1 generation again each other, the gametes would be P, capital, and small p. And you can just check the result of F2 generation crossing PP with PP, capital P, capital P, small p. Then the result would be capital P, 1 flower would be fully purple, dominant purple, homozygous. It will have homozygous allele. That means the alleles capital P are homozygous, similar. 2 flowers that will have heterozygous allele, meaning capital P, small p, uppercase, and lowercase. The last flower will have recessive homozygous allele, small p, lowercase p. This will phenotypically, if we see and consider the phenotypic ratio, it would be 3 purple color to 1 white color. But if we consider and see the genotypic ratio, it is 1 homozygous dominant purple to 2 flowers who has 2 heterozygous alleles and 1 recessive homozygous allele, that is small p. So the ratio, genotypic ratio, would be 1 to 2 to 1. Students, let's try to answer this question. Can you define what monohybrid cross is? Can you define what monohybrid cross is? I'll give you 3 minutes to write down the definition, the detailed definition of this activity, monohybrid cross. I hope that you have tried and answered this question. Very good. Monohybrid cross is a cross, genetic cross between two pure parents differing in a single pair of contrasting characteristics, like the one that you have taken. The two parents were all were pea plants, only they were different in the color of the flower. One parent had purple color, the other parent had white color. So the difference was the color of the flower. So monohybrid cross is a genetic cross between two pure parents differing in a single or one pair of contrasting character or trait. In fact, like the example that you have taken, we have seen that in F2 generation, the phenotypic ratio, phenotypic genotypic ratio was 3:1. We have taken an example of purple color and white color pea plant. We have seen in F2 generation that the pea plant had 3 flowers have purple in color, while 1 flower has only white color. When we see the genotypic ratio, it is genotype, the ratio of F2 generation was 1 to 2 to 1. This means, for example, if you take as color of the flower, 1 flower fully purple, meaning dominant allele, capital P, or uppercase P, while 2 flowers had heterozygous allele, capital P, small p, capital P, small p, while the rest one has recessive homozygous allele. That's 1 to 2 to 1. That's all for today, our students. Next session, we'll continue from the hybrid cross, which is the second genetic cross. Read and come to our next session, the hybrid cross. Goodbye. That's all for today.